Hello everyone, I am Fabian Correa and today I'm going to talk about preserved renal allograft function and the PD-1 pathway inhibitor Nivolumab. The first, what is PD-1 pathway inhibitor? PD-1 is a protein post-immune control found in immune cells called T cells. Normally acts as a type of switch that help prevent T cells attack other cells in the body when binds to PD-L1, a protein in some normal cells.
Every year, nearly 90,000 patients undergo a kidney transplant worldwide. Following the first transplantations in the 50s, allograft survival improved tremendously, with the graft surviving longer and longer. However, there has been no improvement in the survival rate over the past 20 years. And today, 20% of patients lose their new kidney within 5 years of transplantation. These patients must be put back on dialysis once they return to the waiting list, hoping for a new transplant. So we need to extend the survival of transplanted kidneys. To achieve this, patient monitoring after transplantation should be improved, with an individualized understanding of the graft evolution and a personalized treatment. At the Paris Transplant Group, we investigated this by learning from historical records of more than 7,000 patients. For each patient, we collected more than 50 parameters, including histopathological, clinical, and biological data. And among those, we identified 8 parameters in our model that are able to accurately predict the graft survival. We have computed these parameters into an algorithm named iBox. For each transplanted patient, we are now able to predict their probability of graft survival at 3, 5, and 7 years ahead. The iBox has been tested on an external cohort of 4,000 patients and in 3 additional randomized clinical trials. The iBox risk prediction score has been proven to be highly reliable for patients, male and female, from Europe and the US, from different ethnic and social backgrounds, from different allocation systems, covering distinct clinical scenarios, and with different treatment. The iBox is the most validated score available in transplantation. The team at the Paris Transplant Group developed the iBox to guide patient monitoring and made it adaptable to most clinical scenarios, aiming for longer graft survival. Also, for the next generation of clinical trials, the iBox could accelerate the development of novel anti-rejection treatments, help researchers design new studies, and also constitute a valid and early surrogate endpoint. This would accelerate the development of novel anti-rejection treatments. Get further information about iBox in the publication from Alexandre Lupi and his team.
Then infiltrate the tumor microenvironment. The role of the immune system is to protect the body from foreign invaders. Normally, the immune system responds to tumor cells as it does to any foreign invader, by destroying them. T cells play a central role in this immune response to cancer. T cells are primed and activated by dendritic cells within the lymph nodes. They can then infiltrate the tumor microenvironment, which is comprised of tumor cells and other tumor-infiltrating immune cells. Here, the activated T cells detect, bind to, and kill tumor cells. Programmed death ligand 1, or PDL1, is one of the inhibitory ligands that, under normal conditions, help maintain immune homeostasis. However, in many cancers, PDL1 can help tumor cells evade the immune system, particularly when it is expressed in abundance. This occurs when activated T cells release interferon gamma, leading to overexpression of PDL1 on both tumor-infiltrating immune cells and tumor cells. PDL1 interferes with the body's anti-cancer immune response by binding to its receptors B7-1 and PD-1 on the surface of activated T cells, thereby deactivating the T cell's cytotoxic activity. T cells can also become deactivated when PD-L1 expressed on tumor-infiltrating immune cells binds to B7-1 or PD-1.
Applications in medicine. It is important to know that the checkpoint inhibitor used to treat cancer do not work directly on the tumor cells at all. This only remove the brakes on an immune response that has begun but has not yet been working at full power.
Engineered antibodies may be designed to help the body's own defense mechanisms fight diseases. For example, antibodies used in cancer immunotherapy, an emerging therapy option for cancer patients, aim to activate or support the body's immune system in identifying and destroying cancer cells. At Roche Pharma Research and Early Development, we are working on a novel form of engineered antibodies for cancer immunotherapy that we call T-cell bispecifics. The immune system is a very large army of cells organized in different specialized units and regulated by a complex network of messengers. Normally, this defense mechanism protects against foreign organisms, such as viruses and bacteria, as well as cancer cells. Sometimes these mechanisms can fail, and diseases, including cancers, develop. When the immune system fails, cancer immunotherapy helps the immune system get back into action. Among the many types of cells in the immune system, T cells are the most powerful ones that specifically attack cancer cells. Normally, T cells engage in this fight only when they receive a specific message from the cancer cells, presented by an ambassador, the so-called Major Histocompatibility Complex, or MHC. The message is triggered when the ambassador raises specific flags on the cell surface. These flags are called antigens. Only when the ambassador's flags are recognized as foreign do T cells spring into action. This ensures that no healthy cell is attacked by mistake. T-cell bispecific antibodies are basically matchmakers between T cells and cancer cells. The antibodies have two handles that recognize specific markers on the surface of cancer cells, and one handle that is designed to hook to T cells, bringing them in close proximity to the cancer cells. The T cells are then activated to attack the cancer cells. The ambassador role of MHC is not needed anymore, while the need to recognize tumor markers keeps the intervention specific to cancer cells. T-cell bispecific antibodies are designed to be very similar to natural antibodies and to have a lasting action. They can do their job on their own without requiring further technology or modification of the T cells. This is what makes us excited about T-cell bispecific antibodies, a potential new therapeutic option for cancer patients.
Pros and cons. In the first, pros. These drugs have been shown to be helpful in treating various types of cancer. And new types of cancer are being aided as more studies show they are effective. Cons. In the production is slow. Libraries, the cost of production is extremely expensive.
As a scientist, I specialize in cancer immunotherapy. My work, and that of my colleagues, is to figure out ways to stimulate the immune system to kill cancer. To understand this concept, let's talk a little more about our immune system and how it works. Taking a look inside our body, all of us are born with an immune system that acts like the security checkpoint at the airport. Our immune system is like a high-tech full-body scanner system that protects us from things that are harmful. In the immune system, T cells have receptors that scan cells to distinguish between normal cells and abnormal cells, like virus-infected cells. Once detected, abnormal cells are attacked and removed by T cells. T cells also identify these cells for future recognition. The same T cells also play an important role in protecting us from cancer. Cancer cells are normal cells that have mutated. The more mutations cancer cells have, the more easily they are detected. Until recently, we didn't understand why T cells were detecting these cancer cells but not destroying them. But on a closer look, we've made some revealing discoveries. We've learned that one of the ways that cancer cells get past the security system is with the help of a molecule called PD-L1 that is found on the surface of cancer cells. PD-L1 is almost like a disguise that allows cancer cells to remain undetected. PD-L1 basically hacks the T-cell scanning system by jamming the signal to parts of the T-cell system called PD-1 and B7-1. Think of PD-1 as a sensor that's part of the T-cell scanning system, and B7-1 is like an alarm that issues an alert when something is wrong. When PD-L1 hacks the system, it prevents it from doing what it's supposed to do. In other words, it may prevent T cells from destroying cancer cells. Scientists have been working on fixing this problem and have designed molecules to target the PD-L1-PD-1 pathway. You can think of cancer immunotherapy as kind of like a software update for your immune system. Early studies suggest that by targeting the PD-L1-PD-1 pathway, T cells may be able to detect and destroy cancer cells. Let me show you in more detail. T cells have receptors that allow them to recognize cancer. Cancer cells can prevent elimination by turning on PD-L1. PD-L1 deactivates T cells by binding to PD-1 and B7-1. Blocking PD-L1 from binding to PD-1 and B7-1 could prevent PD-L1 from communicating with them. When this happens, T cells may finally be able to respond and kill cancer cells. Clinical trials are currently underway to understand the effects of trying to block the PD-L1-PD-1 pathway in the body. We are committed to studying cancer immunotherapy of cancer.
Issue applied in Ecuador. There is no adequate availability in our market. It is not covered by the state, and the price is very high.
The drugs which are presently in development include drugs from BMS, Nivolumab, Genentech, Roche, MPDL-32808, which is a little bit different from the BMS compound. The BMS compound is a PD-1 antibody. The Genentech is a PD-L1 antibody. And then there's Merck compound MK3475, which is a PD-1 antibody. And in the early studies, the response rate is in the sort of 20% range. And this is in patients with typically heavily pretreated lung cancer, have failed all standard therapies, very resistant to chemotherapy. So a 20% response rate is very encouraging as initial phase 1 data. Many of those patients have had very durable responses. The Nivolumab data was updated at World Lung, and again, in these heavily pretreated patients, the 2-year survival is 24%, which is a very high number for that patient population.
Conclusion. It is an excellent option in the treatment of many types of new plants, and you have an even more promising future against cancer. It is expected that the future will lower the cost of production and can reach more places and be able to be used. Thank you, everyone.